Very good morning, dear students. I hope you all are safe at home and studying hard. So, today's topic is animal cell culture. Let us get started. Animal cell culture or animal tissue culture can be defined as a process of isolation and maintaining the cells, tissues, and nowadays even the organs to some extent of time in vitro. In vitro means outside the body. In vitro, outside the body. So this is very important word, in vitro. So what we are trying to do here in animal cell culture is we are trying to isolate and then we are trying to maintain that isolated cells or isolated tissue and even the organ in vitro outside the body, outside the organism in an artificially controlled microenvironment which is further supported by various aspects. So essentially what I am trying to explain to you is, suppose this is a mice and this mice I want to culture the epithelial cell or fibroblast of this mice. So what I need to do is, first thing in animal cell culture is the isolation. So what I will do is I will dissect the mice and then I will take out the fibroblast or epithelial cell which I want to culture. So the animal tissue culture can be defined as isolation, maintenance of a cell, tissues and even the organs in vitro. In vitro means outside the body. So this is my petri dish. And what I did first, the isolation, I have taken out, I have dissected this mice, I have taken out the cells which I want to culture and I have put this tissues or the cells or the organelles that interest me into the, you can say, the cell culture flask, or this is like a petri dish. So outside, essentially important thing is outside, in vitro. So now these cells will grow and divide outside the body, and so this process is supported. This is called as artificial controlled microenvironment where there is a media and this media is providing all the nutritional requirement. So this media is fulfilling all the nutritional requirement of the cells, like the cell is requiring energy to carry out its metabolic reactions. The cell will require amino acids or the proteins for the source of protein. The cells will require growth factor in order to divide. Besides this, the cell will maintain itself in perfect physiological condition which is maintained, the balanced salt solution. The balanced salt solution is there so that the cell will maintain the physiological pH, the physiological osmolarity, all that is there in the pH. It maintains the cell as it is there in the natural microenvironment. So animal tissue culture, it is a process of isolation, maintenance of a cells or tissues or organ outside the body, which is in artificial controlled microenvironment, which is further supported by cell culture media, and then it is also supported by the advanced equipments like CO2 incubator, which provides the appropriate temperature, physiological temperature to this cell so that it can have the optimum temperature. It will not be stressed out. Then it will maintain the CO2 concentration, where the buffer system of a cell, the bicarbonated buffer system, totally depends on the CO2. So it maintains the 5% of CO2. Then besides this, we have various equipments. So basically what we try to do in the animal cell culture is isolation, maintenance of a cell, tissue or organelle outside in artificial environment which is supported by the media, the cell culture media or tissue culture media, and the equipments like incubators. Why we do this? That part we will see in the application. So different type of, up till now, the different type of cell grown in the culture includes the connective tissue elements, fibroblasts, skeletal tissues, cardiac means the tissue which is related to the heart, the epithelial tissue, liver, breast, skin, kidney, and many different types of tumors are also cultured up till now. So this is most predominant development in the cell culture history. I would like to throw the light on some very important aspects, some very important developments. So the first most important development, although every development is very important, but then the most important, which is the evolution of animal cell culture, I would like to emphasize. So in year 1895, the Ross maintains the embryonic cell culture in saline. So this was the first attempt where the Ross scientists maintained the embryonic. You must be known that the first oncogene SRY, that SRY gene was first isolated by Ross in the chick embryo. So here with relevance to this cell culture, the Ross maintains the first embryonic chick cell in the saline medium. Saline means, you must be knowing saline. Whenever we get weak and we go to doctor inject a saline. Saline is a solution which is having balanced salt concentration plus source of energy, that could be a fructose, glucose, etc. So 1885, the first embryonic chick culture is maintained in saline. Then in 1907, the Harrison first cultured the frog nerve. He dissected the frog, isolated nerve, and cultured it in the lymph clot by a hanging drop method, and observes that nerve fiber in vitro for several weeks. So essentially what he did is he dissect out the frog, isolate that nerve cells. Nerve cells, you must be knowing, it is a cell which resides in our brain, and these are the cells which very seldom divides. These are the cells which rarely divides. So he isolated that nerve cell and he maintained that nerve cell. So he had isolated and then maintained into this biological fluid, we called it as a lymph clot, by hanging drop method, and he observed those nerve fiber for several weeks. So he maintained it outside the body of a frog for several weeks, and that's why so many of them consider him as a father of cell culture. Then in 1911, Levi's made first liquid media. So Levi's made a first liquid media that is made up of seawater, serum, then embryo extract, salt, balanced salt concentrations, and the peptones. 1913, the Carroll introduced a strict aseptic methods, strict aseptic techniques. Aseptic, means absence, septic means bacteria, bacteria-free techniques to culture. Because see, whenever we culture any cell into the media, that media is nothing but a source of nutrition which is going to provide or which is going to fulfill all the nutritional requirement of a cell. That nutritional requirement which is fulfilled by media is itself full of nutrients, and so because of that it can easily catch the infection of a bacteria or the fungus. So in order to maintain it in aseptic condition, one needs to have aseptic techniques, aseptic without bacteria or without any kind of contamination. So that kind of technique was first introduced in 1933 by Carroll. Then in 1916, Ross and Jones introduced a proteolytic enzyme, trypsin. The trypsin, a very important enzyme that you will see while we see the subculturing. Then the next very important development, prominent development is 1952, where the Gay established a continuous cell lines of a human cervical carcinoma known as HeLa cell lines. Nowadays we work on HeLa. This is a cancer cell line. Carcinoma is nothing but a cancer, cervical cancer. The first human cervical cancer of a lady called as Henrietta Lack, that was established, and to your surprise, that cell is still maintained. It is still there, and most of the experiments which is carried out in the cancer research were performed on this HeLa cell lines, which is a source of tremendous insights which is derived from this research experiments. So that HeLa cell lines was first established in 1952, which is still established, and it is a very good source as a which is a very good experimental model to derive the cancer research insights. Then in 1955, Eagle studied the nutritional requirement of a cell. So he studied details, what exactly cell requires to grow, and then he designed a media which is called as chemically defined media. Chemically defined media is a media where all the ingredients is known. 1975, Kohler and Milston produces first hybridoma, the hybridoma monoclonal antibodies, when it is having great application. In the last lecture we have seen fluorescent activated cell sorter. Even in that fluorescent activated cell sorter, it works on the basis of monoclonal antibodies. So that monoclonal antibodies, so essentially what they did is they take out a normal cell and they fuse that normal cell with a B cell cancer, and as B cell is having property to produce an antibodies, so they challenge that B cell in appropriate antigen and they culture that B cell, which is now acquiring a cancerous features. So this discovery was very prominent. This discovery, this Kohler and Milston discovery of 1975 was so prominent that it has produced that antibodies. This technology called as hybridoma technology, this technology has produced a monoclonal antibodies. These monoclonal antibodies are the antibodies which is committed for one particular antigen, and so it is having tremendous applications. Most major applications it is having, which is related to human health. So, and for that reason they were awarded the Nobel Prize in Medicine for such a particular remarkable discovery. Now next comes the animal tissue culture laboratory setup. Now this is a typical laboratory setup for a very large tissue culture. The setup varies according to the lab size, but it is based on the important aspects. So here the different labs have different designs, but basically it is having some principles. So that principles are like here you need to have the complete aseptic area and then different short areas which is dedicated for one particular task. So the animal cell culture laboratory, the foundation was led by Jolly in 1903, and then governing some basic principles, the design may vary according to lab size and lab types. So basically what it is having is here you can see the quarantine room. So whatever new material which arrives in the laboratory first enters into the quarantine room. Besides this, it is having the washing area. Look at this is having separate washing area, and that all the glass wares or plastic wares which is required during this process, it is washed here. Then you can see the sinks are there. Then it is having autoclave room. So autoclave is an equipment which is used to sterile the glasswares or compatible plastic wares. So this is a dedicated autoclave room. And then we have storage room. Besides this is the corridor Then we have single door entry. Nowadays the air curtainers are there. You must have seen that air curtainers in the big shops. Whenever you enter into those shops, for example Haldiram in Nagpur, whenever you enter it, here the curtain is there, and that curtain, when you pass that curtain, it immediately starts and blow air onto you. The main logic behind it is whatever microorganisms you are carrying, it will cut and it will create a negative pressure, and so whatever organisms you are carrying, it will be blown out. And now you enter here. So different laboratories is having different kind of arrangements based on its funding. So once we enter here, the quarantine room is there, then microscope is there. Again, it can be changed according to laboratory size and the requirement. This is a typically large laboratory. So this is a microscope room, and then you enter. This is a main aseptic area. Aseptic area means this area is maintained aseptic condition. So no bacteria, no fungus. So this is perhaps the main area, the aseptic area, and the most important feature of this aseptic area is this area is free of bacterial and fungal infection. One has to keep this area aseptic. One has to keep this area sterile. And how one keep it? One wash or swabs the entire aseptic area. See, this aseptic area is having the incubators, then it is having laminar flow hoods, and then it is these two are very important. And then this is your working area, which is having cell counters, where you count the cell, you count the viability, you access it, and then you have centrifuge. For counting you require a centrifuge. So this is your main working area. This incubators, this laminar flow, and then besides this is the main area. So one has to keep everything sterile here, and so for that what one has to do is one has to swab it. The entire area is swabbed with 70% alcohol, as the alcohol is bactericidal, and so taking 70% alcohol kills almost all the bacteria and also the fungus. So one has to keep it sterile by swabbing it with 70% alcohol periodically. Not only this, besides this, in the weekend one has to fumigate this area. So we have different kind of chemical combinations which is used for the fumigation, which creates a fume, and that fumes kills essentially all the bacteria in this particular environment, this particular area. So the main important thing here is the aseptic area. You have to work here. Here are your laminar flow hoods and the incubators, and so this area has to be sterile. If it is not sterile, then whatever culture you are trying to do will have the contamination. And important thing is the chemicals which is required for the animal tissue culture are expensive chemicals, and so once the experiment is failed, it is a huge loss. It is a loss of time and huge monetary loss, so this animal tissue culture, the aseptic area is there. Besides this, the aseptic area, one has a storage, the freezers, the nitrogen, the liquid nitrogen cans, etc., which is used to store the cells. Okay, so this is the important things that is always there in the animal tissue culture lab. So it is having the washing area, it is having the autoclave, it is having the preparation area. What we do in the preparation area, we prepare the media. And besides this, we have the quarantine room, and then the most important is we have the aseptic area. It is the area where we work, we carry out our work, and so it is maintained the bacteria and fungus free area. Besides this, we have storage. So this is a typical animal tissue culture lab. This is the most important aspects for the success of animal tissue culture, although it is one of the most important aspects, but still the most undervalued aspects, which is undervalued. Now for the first equipment which is required, remember here we are talking about the laminar airflow hoods. Now next is the instruments of animal tissue culture laboratory. So here we will see the important instruments and its basic principles of working and why it is essential, why it is required. So out of this, the first most important is the laminar airflow hoods, which is also called as the biosafety cabinets. So here in this is a photo of a typical vertical laminar flow hood, and this is a horizontal laminar airflow hood. So why it is required in animal cell culture is, here this cabinet is providing totally aseptic region, and this is your actual working region. Whenever you are working, whenever you are copying, whenever you are taking out the cells and culturing it, whenever you are passaging the cells, so all that activities will be done here. And so this region needs to be sterile, and so these airflow hoods, this laminar airflow hood, maintain it as a sterile, because here you can see as compared to room, this is smaller one, and you can easily swab this entire region with 70% alcohol, so that you will have higher efficiency of sterilization. Besides this laminar airflow hood, it is having HEPA. Why we are calling as a vertical laminar airflow hood? Because of the flow of direction of air. So air is flowing here, and besides this, here we have HEPA filters. So this region is having HEPA filters. That HEPA filters is nothing but a high efficiency particulate air filters. And these filters are nothing but small pores which is of lesser than 0.3 micron. And so whenever the air, whatever air is there in this cabinet, this air will circulate inside, and then during this circulation, this air is forced to pass from these HEPA filters. And so if anyhow by accidental some bacteria is still there, that bacteria will be captured in those HEPA filters. So the air is circulated here in vertical direction. So this is the air, this is the air which is shown in the blue color, that is the filtered air, and that air is circulated, then again it is sucked here, and again it is circulated from passing through the HEPA filters, and this is how the process will continue. And so whenever you will work here, you will have whatever air here that will be free of contaminants. And so that is the reason we require this HEPA filters which is there in the laminar airflow hood. Besides this, here you can see the UV light is also, nowadays they are also giving the UV light. So whenever you are working with it, and after the completion of your work, you close this cabinet and then you put on this UV light, and UV light is also, we know, bactericidal activity it has. So UV light is also there. So that is the reason we require a laminar airflow hood. Why? Because it provides a complete sterile microenvironment to carry out the experimental work. It is having HEPA filters. That HEPA filters will filter out all the air which is there. Here you can see the shield is there, glass shield is there. We need to put that glass shield down, and here, from here, we need to insert our hands, and then so that no exterior air will come in. Whatever air is there in the inside will circulate it continuously by getting filtered. Vertical, because the HEPA filter is located at the top, so that the flow of air is from top to bottom, so that becomes a vertical. Horizontal, the horizontal laminar airflow hoods are also there, but we used vertical one. Horizontal, the principle is again the same, but the flow of the flow of the filtered air is horizontal from this direction to this direction. So here you can see from this direction, here the HEPA filters are there, then again the same make is same, the shield is there, small cabinet, so air will flow from this direction to this direction and it will filtered air and it will continuously filter, and so you will have the purified air, contaminant free air here, so that your culture will not have any contamination. So this is the first equipment which is required, laminar airflow hoods. We also called it as a biosafety cabinets. Basically in India we have 4 types. It is universal classification, the biosafety cabinet class 1, class 2, class 3, and nowadays we even have class 4 and class 5 also. It is totally based on the safety. Okay, so this is the simplest form of the biosafety cabinet. Whatever we are using. If you have visited our cell culture facility at Department of Zoology, we are using this class 2 biosafety facility, where you can have this closed compartment and then jubilite is there. When this kind of facility is there in the various national laboratories, so if you are working with the highly infectious agents like HIV virus, or even nowadays the COVID is there, which is supposed to infect most of the doctors and the masses. Why? Because it is having high capacity to infect, and so when you want to work for the vaccine of such viruses or other deadly viruses like HIV or HTLV1, such work requires a high degree of protection. And so here you can see you will not have any kind of glass shield which is like here. Here there is everything is there. It is a here you can see you have to insert your hands from this, the rubber wall sort of thing is there, and you have to insert your hands from this, and then you have to work out. The HEPA filters are there, but whatever outlet which is coming out from this biosafety level 3 and onwards, that is 4 and 5, whatever air which is circulating there, then that air is passed into incinerator. The incinerator means very high temperature, and so the infectious agents that you are working on, even if accidentally it gets into that air is passed into the incinerator, and so that is essentially neutralize that threat. And so for that reason we have class 3, class 4 facility. The class 3 is there in the NCCS Pune, and class 4 is also there in NCCS Pune and National Institute of Immunology, New Delhi. So these are the biosafety cabinets that we have up till now. The second most important equipment that is required for the success of animal tissue culture laboratory is CO2 incubators. Now these are the special type of incubators where it is associated with a CO2 cylinder. So here it is a rough sketch of CO2 incubators which is having the control panel. The important thing is why we require the CO2 incubators, because it provides the ambient temperature which is the optimum temperature for the cells. At physiological condition, we have the human being is having 37 degrees Celsius of body temperature, and so we have to maintain it precisely. So this cabinet provides that appropriate temperature which is set. We can set that temperature from this control panel, and it will maintain the temperature as it is, which is there, which is a physiological temperature, of course, because if the temperature somehow increases, then it will cause the stress to the cells, and then the cells response will be different, right? Cells may die. So in order to avoid this, we need to have the CO2 incubators. This CO2 incubator is provided it provides three important things. The first, it provides the appropriate physiological temperature. So we can set it, as it is an incubator, it will maintain that temperature. The second important thing it provides is CO2, and that to the 5% CO2, because in the media we have added, you must have studied the buffer system, the bicarbonate buffer system, and in order to maintain buffer system, to maintain the pH appropriately, because we have to maintain the pH, we have to maintain the cell, and cell will grow optimally in its optimum pH, and so we have to maintain this pH, right? This pH has to be maintained, and for the maintenance of a pH at particular range we need a buffer system. So here we have a bicarbonate buffer system that you have already studied in the chemistry, and most important aspects of that bicarbonate buffer system is the CO2 concentration. The CO2 concentration, and so that CO2 concentration is provided to that particular cell with the help of this CO2 incubator. So this incubator is having pipe, and that pipe is joined to the this is the CO2 cylinder, and then CO2 cylinder, it is having a regulator. Here this is a regulator, and this pipe is joined to the regulator, and so this is the regulator. Okay, this is CO2 cylinder, this is regulator, and this is the connection. So this CO2 cylinder, this regulator, we release appropriate amount of CO2 into it, and through this digital board we control it at 5% CO2. So basically we maintain the 5% of atmospheric CO2 in this cabinet. Why? Because the buffer system, more specifically bicarbonate buffer system, requires the 5% of CO2 here, so that the media will maintain its pH. That is the reason why we have the 5% CO2 buffer, CO2 to maintain the buffer system of bicarbonate. The third thing it provides is the humidity. So nowadays what we used to do is we used to put a petri dish which is full of water, which is full of sterile water here, right? So water is also autoclave, so we reduces the probability of infection, contaminant through this water. So that water is also autoclaved, and we put that water into this petri dish, and we put that petri dish into this cabinet, so that at this particular temperature, the temperature what you have said, it will provide the optimum humidity, and cell will have the optimum humidity, and cell will maintain itself. So this is a typical photo of a CO2 incubators, where you can see shelf here, then you have meter reading which provides the temperature. Outside also you will have meters, digital meters, which provides the temperature and the mechanism to set it. And then nowadays we have also light. So one more glass shield is there, and here we put that autoclave water to maintain the humidity, and then from back side we maintain the CO2. So the small petri dish or T25 flask we keep into this frames, and it will maintain, we close it, and it will maintain the appropriate temperature. Whatever temperature you have set, it will maintain the CO2 concentration of 5%, whatever you have set, and it will maintain the humidity, and cell, it will mimic the, it will try to mimic the natural condition where cell resides originally. The third instrument, the third most important instrument which is required for the cell culture is of course the microscope. But here we use different kinds of microscope, and this kind of microscope is called as a inverted microscope. Inverted microscope, right? So it is same, the function is same. The small objects you have to observe it, so you need to magnify it, and for that reason you require a microscope. But here we required a special inverted microscope. Inverted, the simple meaning of inverted is opposite. Why we called it as an inverted microscope? Because in conventional compound microscope the source of light was at the bottom and the object, the eyepiece, as is at the top, right? So this is the source of light. In normal conventional compound microscope, the source of light is there, then source of this is your stage, and then this is the condenser, this is the object that you want to magnify, and then these are your objective lenses, and then here is your eyepiece, right? Why we call this kind of microscope is inverted is because here the source of light, look at this is the source of light. The source of light here is from top, and then you have stage, this is the source of light, and then there is the condenser, and then here is your object or specimen that you want to magnify, that here the stage is, and then you have the objectives at the bottom. So this is the source of light which is passing through, and then it is getting into objective, and then that light is deflected by mirror into another mirror, and then it will create the image. This is your eyepiece. Though simple, it's very simple. Why we called it as an inverted microscope? Because the source of light is opposite. In normal, the source of light is at bottom, but here the source of light is at top. Then here we have condensers. In normal conventional compound microscope, the objectives resides here, but in this inverted microscope the objective resides at bottom, inverted position. Why we need an inverted microscope? Because this is a petri dish culture, and this petri dish is having cells. These are the cells. Important thing is this cells is attached at the bottom, and then you have fluid that is called as media at the top of that. That is the reason you need to have the source of light from the top, right? And you have the objective, so that you will have closer and appropriate views of the cell. It will not be appropriate if this arrangement is like normal compound microscope, that is at the top. When this objective will be at top, it will not capture it appropriately, as the media will be there in between, and so no proper cellular anatomy can be studied here. That is the reason we require an inverted microscope. The second thing, we required a plastic wares and autoclave, micropipettes, etc. So again, these are the smaller instruments, but again very important instruments. This is called as an autoclave. This is called as a micropipette, that means you have already used in the microbiology. And these are the plastic wares. This typical plastic ware is called as T25 flask. This is made up of plastic. So the first is the autoclave. This autoclave, you can, if you want me to give an analogy of autoclave, this autoclave is nothing but a pressure cooker. It is having meter, then it is having the safety valves, the two safety valves are there, and then it is having joints which closes this lid. It is having digital meter. So what basically we do here is we add a water here into this autoclave. We open this lid, we add a water, and whatever glasswares which is already cleanly washed, which is wrapped around with a paper and put into this autoclave. All the stoppers are closed, all the screws are closed appropriately, and this meter is set, and then we put on. The water is poured, the glasswares or compatible plastic wares that we need to autoclave is put in. We close the lid, we tighten the screw and we put it on, we set it. So what happens is, as the coil is there, this coil gets heated, and because of that heat, whatever water we have added in the bottom, it gets boiled. And we all know the boiling temperature of water is 100 degrees Celsius. So when it reaches 100 degrees Celsius, the water starts boiling and get converted into vapors. As the lid is closed and valves are there, so it creates a water vapor, but then that water vapor is not having space to leave it, causing the increase in the pressure. And so the important principle that you should study here is, whenever the water is boiled, it hardly achieves the temperature. Whenever you boil the water, it achieves the maximum temperature of 100 degrees Celsius. But then we have some fungal spores which survives 100 degrees Celsius because of their covering. And so we need a higher temperature. And for that, the water can be boiled at 100 degrees Celsius, it produces vapor. But if you don't allow that vapor to leave, then the temperature inside this vessel can increase up to 121 degrees Celsius. Why? Vapor pressure. Because the pressure is there, you are increasing the pressure, the water is still boiling, it causes the rise of a temperature up to 121 degrees Celsius. And so we use at 121 degrees Celsius for 30 minutes. If you keep any glassware at this temperature for 30 minutes, it will cause all the bacteria and microorganisms to die. Even the fungal spores will be dead at this temperature. And so that is the reason we used autoclave. Whatever media that we are using, we first do autoclaving. Whatever glasswares that we are using, we first do it autoclave. And even the water we are using in the media, we autoclaved it. We kill all the bacteria and the fungal spores using this high temperature. So it is just a pressure cooker. The function of this safety valve here is, whenever the pressure increases and you still keep it on, the temperature is increasing, the pressure is increasing, and if you have not opened it or not turned off it, what it will cause? It will cause this safety valve to release the pressure exactly like the pressure is released in the pressure cooker through whistle. So it is a safety valve. It prevents the blast of this autoclave. So whenever the pressure exceeds, it opens that safety valve and that steam will come out of it, and again it gets closed. Very similar to that happens in the pressure cooker. So that is the autoclave we require. The second thing we require here is the micropipette. Because we want to work at cellular level, and so essentially the most prominent requirement is to work at micro level. All the chemicals, the media, the dyes, all that is required to work at micro level, and for that we requires a micropipette. So here you can see the meter is there, the reading is there. By rolling this screw you can adjust it, and then by pressing it is simply working on the phenomena of a The second equipment is a micropipette. Here this is a micropipette. I guess you have already used this micropipettes. Micro means small, pipette. So pipette we use to aspire the solution, right? So we need to work at cellular level, and cellular level requires the chemicals, the dyes, the solutions at micro level, and for that the conventional pipette cannot be used. we need a specialized pipette called as micropipette. The whole principle of this micropipette is based on that calibrated spring. Here you can see this knob, you circulate this knob and you change the settings here according to your requirement. 1 microliter is there. If your requirement is 1 microliter, you set that 1 micrometer by rolling this knob, and then you aspire solution by pressing this knob. You press this knob, then we have tips here. So you aspire the solution by releasing this knob. The knob will come to its original position based on the spring, mechanism of spring, and appropriate, that is 1 microliter of solution will be aspired in this tip. Now you put that solution into appropriate vial, and then after that you can discard this tip. So that is why we require a micropipette. And lastly, we require a plastic wares. Here these are special culture flask which is designed for the animal tissue culture. It is made up of negatively charged plastic. Here the cells are there, the cells get added to the bottom. The area of this flask is measured. You add appropriate, usually if it is T25, based on its volume it is T25, T50 and T75 also. This one is T25. For T25 we added 2.5 ml of a media. Beside this, the cap is there, the neck is there, the neck is slightly towards upside, and then cap is there and small perforations are there in the cap, so that whatever CO2 concentration you set in the incubator, usually we set a CO2 concentration of 5%. So whatever CO2 concentration you set in that CO2 cabinet, it will get diffused inside, because the bicarbonate system is working in this media. So we need to have that CO2 inside this. But if you keep this cap open, it may cause the contamination. So in order to rectify that problem, the small perforations are there in the cap. Here you can see those perforations are visible, and those perforations are for reason that the CO2 can enter from these perforations into here, and it helps in that maintenance of pH in bicarbonate of the system. So that is reason we need autoclave, micropipettes and glasswares. Here the primary culture, secondary culture, container cell lines and passaging we will see in the next lecture. Thank you.